Our topic is the use of animals for human benefits, and we will be focusing on the animals used in laboratory experiments.
Mice, rats, dogs, cats, rabbits, guinea pigs, monkeys, and other animals are all used for experiments. These animals are used for biology lessons, medical training, chemical, drug, food, and cosmetic testing.
There are two sides to using animals for human benefits, the pros and the cons, which will be presented to help others form a better educated opinion.
The four main areas that will be covered are cures and treatments, alternate testing methods, animal compared to humans, and benefiting animals.
First up are cures and treatments. The California Biomedical Research Association states that almost every medical breakthrough in the last 100 years was because of research using animals. Experiments with dogs who had their pancreas removed led directly to the discovery of insulin, which is critical to saving people who have diabetes. Chris Abbey, director of the University of Texas MD, states, "We wouldn't have a vaccine for hepatitis B without chimpanzees." And for the opposing con side,
Animal tests may mislead researchers into overlooking potential cures and treatments for humans. There are some things that are deadly to animals, but they're helpful to humans. An example of this is aspirin, which is dangerous to some animal species. When it was being tested on animals, the results showed negative and harmful effects on the animals. With these results, researchers almost did not let aspirin be tested on humans. To humans, aspirin is a very helpful drug. It is used by many patients who have transplants to keep their body from rejecting new organs. There could be humans suffering because certain drugs will not be used on humans because of their negative effects on animals.
alternate testing methods.
It is difficult to replace the use of living animals in research with alternative methods. Studying cells in a petri dish, while sometimes useful, does not provide the opportunity to study the interrelated process. Evaluating a drug for side effects requires a circulatory system to carry medicine to different organs. Conditions such as blindness and high blood pressure cannot be studied in tissue cultures. Even the most powerful supercomputers are unable to accurately stimulate the workings of complex organs such as the brain.
The opposing con side. There are alternate testing methods that will eliminate the use of animals in the lab. Using vitro testing, which is studying cells from a petri dish, is an alternate to using live animals. Microdosing is another alternate, which is administering a small dose to cause adverse reactions in humans, whose blood will then be analyzed. Artificial human skin, like epiderm, is commercially available, which will produce a more human reaction than something tested on animal skin. Another option is to use a computer model that will stimulate the effects from the drugs. Animals compared to humans.
Chimpanzees share 99% of DNA with humans, and mice are 98% genetically similar to humans. All mammals are descended from the common ancestors and have the same set of organs. Because of this, humans and animals share many illnesses. Dogs and humans share cancer, diabetes, and ulcers. Rabbits and humans share emphysema, and cats and humans share visual impairments.
The opposing con side. Animals are different from humans, so therefore make bad testing subjects. 92% of experimental drugs that are safe and effective on animals fail in human clinical trials because they are dangerous and do not work on the human body. There are autonomic, metabolic, and cellular differences in animals, which makes them poor models for humans. Paul Furlan, professor of clinical neuroimaging at Aston University, states, "It's hard to create an animal model that even equates closely to what we're trying to achieve in the human." Another professor, Thomas Harting, professor of evidence-based toxicology at Johns Hopkins University, argues that alternatives to animal testing because we are not 70-kilogram rats. Benefiting animals. If vaccines were not tested on animals, millions of animals would have died from rabies, distemper, and canine parvovirus. Animal testing has also been instrumental in saving endangered species from extinction. Koalas, California condor, and the tamarines of Brazil are all examples of endangered species that were saved. Treatments for animals developed using animal testing also includes pacemakers for heart disease and remedies for glaucoma and hip dysplasia. Opposing cons side.
Drugs that pass animal tests are not necessarily safe. The sleeping pill thalidomide was tested on animals and did not cause birth defects in the animals unless they took extreme doses. Over 10,000 human babies were born with severe deformities. The drug Vioxx was supposed to be a protective heart drug, which was tested on mice and was very successful. However, it killed 27,000 humans through cardiac death before it was removed from the market.
So how does one feel for animals for human benefit after receiving all this information? Here's a poll on how people feel about scientific experiments on animals. Are they necessary and/or acceptable? With this new information, one is able to finally determine a very well-educated opinion about animals being used for human benefit.